conformation. A spatial arrangement of substituent groups that are free to assume different positions in space, without breaking any bonds, because of the freedom of bond rotation. Native conformation. The biologically active conformation of a macromolecule. Hydrophobic interaction. The association of nonpolar groups or compounds with each other in aqueous systems driven by the tendency of the surrounding water molecules to seek their most stable state. Solvation layer. When water surrounds a hydrophobic molecule, the optimal arrangement of hydrogen bonds results in a highly structured shell. Peptide group. A peptide bond is a covalent chemical bond formed between two molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule, causing the release of a molecule of water. Hence the process is a dehydration synthesis reaction, and usually occurs between amino acids. Ramachandran plot is a way to visualize backbone dihedral angles psi against 5 amino acid residues in protein structure. Secondary structure. The local spatial arrangement of the main chain atoms in a segment of a polypeptide chain, also applied to polynucleotide structure. Alpha helix. A helical conformation of a polypeptide chain, usually right-handed, with maximal intra-chain hydrogen bondings, one of the most common secondary structures in proteins. Beta conformation. An extended, zigzag arrangement of a polypeptide chain, a common secondary structure in proteins. Beta sheet. Hydrogen bonds form between adjacent segments of polypeptide chain. The zigzag polypeptide chains can be arranged side by side to form a structure resembling a series of pleats. Beta turn. A type of protein secondary structure consisting of 4 amino acid residues arranged in a tight turn so that the polypeptide turns back on itself. Circular dichroism spectroscopy. Structural symmetry in a molecule gives rise to differences in absorption of left-handed versus right-handed plant polarized light. Measurement of this difference is called tertiary structure. The three-dimensional conformation of a polymer in its native folded state. Quaternary structure. The three-dimensional structure of a multi-subunit protein, particularly the manner in which the subunits fit together. Fibrous proteins. Insoluble proteins that serve a protective or structural role contain polypeptide chains that generally share a common secondary structure. Globular proteins. Soluble proteins with a globular shape. Alpha-keratin. Found mostly in mammals, these proteins constitute almost the entire dry weight of hair, wool, nails, claws. This protein has evolved for strength. It is a right-handed alpha helix. Collagen. Like alpha-keratins, it has evolved to provide strength. It is found in connective tissues such as tendons, cartilage, the organic matrix of bone. It is a unique secondary structure, quite distinct from alpha helix. It is left-handed and has 3 amino acid residues per turn. Silk fibroin. Its polypeptide chains are predominantly in the beta conformation, rich in ALA and glue residues, permitting a close packing of beta sheets and an interlocking of R groups. Protein Databank. An international database that archives the data describing the three-dimensional structure of nearly all macromolecules for which structures have been published. Motif. A distinct folding pattern for elements of secondary structure, observed in one or more proteins. A motif can be simple or complex, and can represent all or just a small part of a polypeptide chain, also called a fold or supersecondary structure. Domain. A structural unit of a polypeptide. Domains may have separate functions and may fold as independent compact units. Protein family. Proteins with significant similarity in primary structure and, or similar tertiary structure and function are said to be in the same blank. Multimer. A multi-subunit protein. Oligomer. A short polymer, usually of amino acids, sugar or nucleotides. The definition of short is somewhat arbitrary and usually fewer than 50 subunits. Protomer. A general term describing any repeated unit of one or more stably associated protein subunits in a larger protein structure. If a protomer has multiple subunits, the subunits may be identical or different. Symmetry. Subunits can be superimposed by rotation about a single axis. Denaturation. Partial or complete unfolding of the specific native conformation of a polypeptide chain, protein, or nucleic acid such that the function of the molecule is lost. Molten globule. The state resulting from hydrogen collapse may have a high content of secondary structure, but many amino acid side chains are not entirely fixed. The collapsed state is often referred to as molecular chaperone. Proteins that interact with partially folded or improperly folded polypeptides, facilitating correct folding pathways or providing microenvironments in which folding can occur. HSP70. More abundant in cells stressed by elevated temperatures, these proteins bind to regions of unfolded polypeptides that are rich in hydrophobic residues, preventing inappropriate aggregation. These chaperones thus protect both proteins subject to denaturation by heat and new peptide molecules being synthesized. This protein also blocks the folding of certain proteins that must remain unfolded until they can be translocated across a membrane. Chaperonin. These are elaborate protein complexes required for the folding of some cellular proteins that do not fold spontaneously. Amyloid. Insoluble fibrous protein aggregates sharing specific structural traits. Prion. A normal constituent of brain tissue in all mammals, its role is not known in detail, but it may have a molecular signaling function. Pro-residues in the CI's configuration. Accommodate turns well. Aromatic residues play an important role in stabilizing amyloid fibrous. What is the primary structure of a protein? Its amino acid structure. Proteins typically consist of how many amino acids? From 50 to 2000. What is the secondary structure of a protein? The three-dimensional structure that results from a regular pattern of hydrogen bonds between the NH and the CO components of amino acids. What is the tertiary structure of a protein? The three-dimensional structure that results from bonds between the R groups of amino acids. What is the highest level of structure that an individual protein can attain? Tertiary structure. What is quaternary structure of a protein? The structure that results when one or more polypeptide chain bond together to form a single macromolecule. What is the final three-dimensional structure of a protein determined by the amino acid sequence of a protein? What is the linkage that joins amino acids in a protein called A peptide bond or an amide bond. Where exactly do peptide bonds form? Between the alpha-carboxyl group of one amino acid and the alpha-amino group of another amino acid. What is the formation of a dipeptide from two amino acids accompanied by the loss of a water molecule? What does the equilibrium of this reaction lie on the side of under most conditions? Hydrolysis rather than synthesis. Hence, the biosynthesis of peptide bonds requires what? An input of free energy. Why are peptide bonds quite stable kinetically? Because the rate of hydrolysis is extremely slow. What is each amino acid within a polypeptide chain known as? A residue. Do polypeptide chains have directionality? Yes. Why? Because its ends are different. How? There is an alpha amino group at one end and an alpha carboxyl group at the other. By convention, which end is taken to be the beginning of a polypeptide chain? The amino end. What are the two main components of a polypeptide chain? 1. Main chain, backbone, repeating part. 2. Side chains, variable part. What is the polypeptide bond rich in? Hydrogen bonding potential. Why? Because each residue contains a carbonyl group and an amino group. The carbonyl group is a good hydrogen bond acceptor. The amino group is a good hydrogen bond donor. What is the largest protein? Titin. How many amino acids does it consist of? Almost 27,000. What is its role in the body? Serves as a scaffold for the assembly of the contractile proteins of muscle. What are peptides made of? Small numbers of amino acids known as oligopeptides are simply peptides. In some proteins, the linear polypeptide chain is covalently cross-linked. What are the most common links? Disulfide bonds, to conform between cysteine residues in the same polypeptide chain, or they can link two separate chains together. What are disulfide bonds formed by? The oxidation of a pair of cysteine residues. What is the resulting unit of two linked cysteines known as cysteine? Are non-disulfide cross-links derived from other side chains ever present in proteins? Rarely. Work on what hormone should add a protein has a precisely defined amino acid sequence. Insulin. List 4 reasons why knowing amino acid sequences is important. 1. They determine the three-dimensional structures of proteins. 2. Knowledge of the sequence of a protein is essential to elucidating its mechanism of action. 3. Sequence determination is a component of molecular pathology. 4. The sequence of a protein reveals much about its evolutionary. Describe the shape of a peptide bond. Peptide bonds are essentially planar. Thus, for a pair of amino acids linked by a peptide bond, how many atoms lie in the same plane? Six. List these 6. 1. Alpha carbon atom and CO group of the first amino acid. 2. NH group and the alpha carbon atom of the second amino acid. What else is significant about the peptide bond? It has considerable double bond character. Why? Owing to resonance structures, the electrons resonate between a pure single bond and a pure double bond. What does this partial double bond character prevent? Rotation about the peptide bond. Or peptide bonds charged? No. What does this allow for? Amino acids to form tightly packed globular structures that would otherwise be inhibited by charge. Repulsion. What two configurations are possible for a planar peptide bond? 1. Trans configuration. 2. Cis configuration. Describe the location of the alpha carbon atoms in the trans configuration. The two alpha carbon atoms are on opposite sides of the peptide bond. Describe the location of the alpha carbon atoms in the Cis configuration. The two alpha carbon atoms are on the same side of the peptide bond. Are most peptide bonds in protein Cis are trans? Trans. Why? There are steric clashes between R groups in the Cis configuration. Are the bonds between the amino group and the alpha carbon atom and the carbonyl group and the alpha carbon atom single or double bonds? pure single bonds. What does the freedom of rotation that results allow for? Allows for proteins to fold in many different ways. What can the rotations about these bonds be specified by? Torsion angles. What is the angle of rotation about the bond between the nitrogen atom and the alpha carbon atom? Called phi. What is the angle of rotation about the bond between the carbonyl carbon atom and the alpha carbon atom? Called psi. Does a clockwise rotation about either bond as viewed toward the alpha carbon atom correspond to a positive or negative value? Positive value. What do the phi and psi values determine? The path of the polypeptide chain. Are all combinations of phi and psi possible? No. Why not? Due to steric clashes between atoms. What is steric exclusion? The fact that two atoms cannot be in the same palsy at the same time. What does steric exclusion restrict? The number of possible peptide conformations. Thus, this makes it a powerful organizing principle. What four regularly folded segments comprise a proton secondary structure? 1. Alpha helix, 2. Beta pleated sheet, 3. Turns, 4. Loops. What are alpha helices, beta pleated sheets, turns and loops formed by? A regular pattern of hydrogen bonds between the peptide NH and CO groups. Describe an alpha helix. Rod-like, tightly coiled, side chains extend outward in a helical array. What are alpha helices stabilized by? Hydrogen bonds between the NH and CO groups of the main chain. The CO group of each amino acid form a hydrogen with which NH group? The NH group of the amino acid that is situated 4 residues ahead in the sequence. With an alpha helices, do a large number or a small number of functional groups participate in hydrogen bonding? A large number of groups, 2 except for amino acids near the ends of an alpha helix. All the main chain CO and NH groups are hydrogen bonded. How is each residue related to the next one? by a rise, translation of 1.5 angstroms. This gives how many amino acid residues per turn of helix. Minus 3.6 amino acid residues. Thus, amino acids spaced blank and blank apart in the sequence are spatially quite close to each other. Thus, amino acids spaced 3 and 4 apart in the sequence are spatially quite close to each other. What is the pitch of an alpha helix? The length of one complete turn along the helix axis. How do you calculate the pitch of an alpha helix? Pitch equals translation x residues per turn equals 5.4a. What does screw sense refer to? The direction in which a helical structure rotates with respect to its axis. What are the two types of screw sense? One, right-handed, two, left-handed. How can you identify a right-handed screw sense? If viewed down the axis of a helix, the chain turns in a clockwise direction. How can you identify a left-handed screw sense? If viewed down the axis of a helix, the chain turns in a counterclockwise direction. Are right-handed or left-handed helices more energetically favorable? Right-handed helices. Why? Because there are fewer steric clashes between the side chains and the backbone. As a result, essentially all alpha helices in proteins are right-handed. Can all amino acids be readily accommodated in an alpha helix? No. What two main types of amino acids don't like to form alpha helices? One, amino acids that possess side chains with branching at the beta-carbon atom. Two, amino acids that possess side chains with hydrogen bond donors or acceptors in close proximity to the main chain, where they compete for main chain NH and CO groups. Which amino acids possess side chains with branching at the beta-carbon atom? One, Choline. Two, threonine. Three, isoleucine. Which amino acids possess side chains with hydrogen bond donors or acceptors? One, serine. Two, aspartate. Three, aspartate. Why is proline a helix breaker? 1. It lacks an NH group. 2. Its ring structure prevents it from assuming the phi to fit into an alpha helix. Describe how the alpha helical content of proteins ranges. Wheatley, from none to almost 100%. What percentage of all soluble proteins are composed of alpha helices connected by loops and turns? About 25%. Single helices are usually how long? Less than 45 angstroms. What is a beta-pleated sheet composed of? Two or more polypeptide chains called B strands. A beta strand is almost fully blank rather than being tightly coiled as in the alpha helix. A beta strand is almost fully extended rather than being tightly coiled as in the alpha helix. What is the distance between adjacent amino acids along a beta strand? Approximately 3.5 angstrom. What is the distance between adjacent amino acids along an alpha helix? Approximately 1.5 angstroms. How is a beta sheet formed? By linking two or more beta strands lying next to one another through hydrogen bonds. Describe the two directions in which adjacent chains in a beta sheet can run. One, in opposite directions, anti-parallel. 2, in the same direction, parallel. How many strands can come together in a beta sheet? Typically 4 or 5, as many as 10 or more. Beta sheets can be purely blank, or blank. Beta sheets can be purely parallel, purely anti-parallel, or mixed. Beta sheets can be mostly blank, but some adopt the blank shape. Beta sheets can be mostly flat, but some adopt a twisted shape. What structural elements are used to accomplish reversals in the direction of polypeptide chains? Reverse turns and loops. In what part of proteins do turns and loops lie? On the surfaces. Loops and turns exposed to an aqueous environment are usually composed of amino acids with what type of R groups? Hydrophilic R groups. What are two common types of fibrous proteins? 1. Alpha-keratin, 2. Collagen. What is alpha-keratin a component of? Wool and hair. What does it consist of? Two right-handed alpha helices intertwine to form a left-handed superhelix called a coiled coil. Alpha-keratin is a member of a superfamily of proteins referred to as coiled coil proteins. Name 3 types of interactions that help to cross-link the two helices with an alpha-keratin. 1. Van der Waals forces. 2. Ionic interactions. 3. Disulfide bonds. What is the most abundant mammalian protein? Collagen. Where is collagen found within the human body? Skin, bone, tendon, cartilage, and teeth. Describe the structural composition of collagen. Contains 3 helical polypeptide chains, each nearly 1,000 residues long. What amino acid appears at every third residue? Glycine. What 3 residue pattern recurs frequently? Glycine, proline, proline. What are absent in this type of helix? Hydrogen bonds within each peptide chain. How then are the helices stabilized? By steric repulsion of the pyrrolidine rings of the proline residues. Describe how the 3 helices interact with each other. 3 strands wind around each other to form a superhelical cable. How is the cable stabilized? By hydrogen bonds between the strands. What groups do the hydrogen bonds form between? The peptide NH groups of glycine residues and the CO groups of residues on the other chains. The inside of the triple-stranded cable is very blank. The inside of the triple-stranded cable is very crowded. How does this explain why glycine has to be present at every third position on each strand? The only residue that can fit in an interior position is glycine. The replacement of glycine by other amino acids leads to what disease? Osteogenesis imperfecta, brittle bone disease. What does this replacement lead to? Delayed and improper folding of collagen. What does the accumulation of defective collagen result in? Cell death. What is hydroxyproline? Modified version of proline with a hydroxyl group replacing a hydrogen atom in the pyrrolidine ring. Why is hydroxyproline so crucial in the body? It is essential for stabilizing collagen. Why is vitamin C required for the formation of stable collagen fibers? Because it assists in the formation of hydroxyproline from proline. Specifically, it is required for the continued activity of prolyl hydroxylase, which synthesizes hydroxyproline. What does less stable collagen result in? Scurvy. Describe how prolyl hydroxylase is activated. Pha2 sits on prolyl hydroxylase. Pha2 activates O2. Prolyl hydroxylase catalyzes the conversion of hydroxyproline. What happens when Pha2 is oxidized to form Pha3? Prolyl hydroxylase becomes inactivated. How is the enzyme made active again? Vitamin C reduces Pha3. Thus, vitamin C ascorbate serves as an antioxidant. What does tertiary structure refer to? The spatial arrangement of amino acid residues that are far apart in the sequence into the pattern of disulfide bonds. What is this level of structure the result of? Interactions between the R groups of the peptide chain. What is the first protein to be studied in anatomic detail? Myoglobin. Myoglobin is an example of what type of protein? A globular protein. Describe the characteristics of a globular protein. Compact three-dimensional structure, water-soluble. What type of proteins perform most of the chemical transactions within the cell? Globular proteins. What is the role of myoglobin in the body? Facilitates the diffusion of oxygen from the blood to the mitochondria. Where is myoglobin predominantly found? In heart and skeletal lamsuckle. What does the capacity of myoglobin to bind oxygen depend upon? The presence of iron-binding hemo groups. About 70% of the main chain of myoglobin forms what type of structures? Alpha helices. Why is myoglobin asymmetric? Due to the complex folding of its main chain. What type of residues does the interior of myoglobin consist of? Almost entirely of nonpolar residues. What are the only polar residues on the interior? Two histidine residues. Why are they present in that location? They play critical roles in binding both heme and oxygen. What does the outside of myoglobin consist of? Both nonpolar and polar residues. In an aqueous environment such as the interior of the cell, protein folding is driven by The hydrophobic effect. What is the hydrophobic effect? The strong tendency of hydrophobic residues to avoid contact with water. What environment does an unpaired NH/CO group FO the main chain prefer? Water. What is the only way to bury a segment of the main chain in a hydrophobic environment? To pair all the NH and CO groups by hydrogen bonding. How is this pairing neatly accomplished? In an alpha helix or a beta sheet. Why does the set of 20 amino acids differ so much in size and shape? They provide a palette of shapes that can fit together tightly to fill the interior of a protein neatly and thereby maximize van der Waals interactions. Some proteins that span biological membranes are the exceptions that prove the rule. Why? Because they have the reverse distribution of hydrophobic and hydrophilic amino acids. Provide an example. Porins are found on the outer membranes. Some bacteria, porins are covered on the outside largely by hydrophobic residues that interact with the hydrophobic environment of the cell membrane. In contrast, the center of the protein contains many charged and polar amino acids that surround a water-filled channel running through the middle of the protein. List two types of motifs, supersecondary structures. Helix. Turn helix, domains connected by a flexible polypeptide chain. List all of the bonds that form a proton's tertiary structure. 1. Disulfide bonds. 2. Hydrophobic interactions. 3. Hydrogen bondings. 4. Salt bridges. 5. Van der Waals interactions. 6. Ionic bonds. What does quaternary structure refer to? The arrangement of protein subunits and the nature of their interactions. It can be as simple as two identical subunits or as complex as dozens of different polypeptide chains. List the different type SOF interactions among subunits of proteins. 1. Disulfide bonds. 2. Hydrophobic interactions. 3. Hydrogen bondings. 4. Salt bridges. 5. Van der Waals interactions. 6. Ionic bonds. 7. What is the simplest sort of quaternary structure? A dimer consisting of two identical subunits. Chaotropic agents such as urea disrupt what type of bonds in proteins? All of the non-covalent bonds in a protein. What agent can be used to cleave disulfide bonds? A sulfitero reagent such as beta-mercaptoethanol. What happens when ribonuclease is treated with urea and beta-mercaptoethanol? Converted to a randomly coiled polypeptide devoid of enzymatic activity. Protein is said to be denatured. What happens when ribonuclease is freed of urea and beta-mercaptoethanol by dialysis? Protein slowly regains its enzymatic activity, all of the physical and chemical properties return to normal, spontaneously refolds into a catalytically active form with all of the correct bonds reforming. Does refolding always proceed efficiently? No. Why not? Unfolded protein molecules can become tangled up with one another to form aggregates. How does a protein make the transition from an unfolded structure to a unique conformation in the native form? List 2 options. 1. All possible conformations are tried out to find the energetically most favorable one. 2. Proteins follow at least a partly defined folding pathway consisting of intermediates between the fully denatured protein and its native structure. What is the essence of protein folding? The tendency to retain partly correct intermediates. Why is there this tendency? Because partly correct intermediates are more stable than unfolded regions. What is the criterion of correctness in protein folding? The total free energy of the folding intermediate. What is sometimes used to help visualize the folding of proteins? Folding funnel, energy landscape. What does the breadth of the funnel represent? All possible conformations of the unfolded protein. What does the depth of the funnel represent? The energy difference between the unfolded and the native protein. What does each point on the surface represent? A possible three-dimensional structure and its energy value. What are the 3 main model folding pathways that have been proposed? One, local interactions, secondary structure, to long-range interactions, tertiary structure. 2, the hydrophobic effect brings together hydrophobic amino acids that are far apart in the sequence. 3. Nucleation condensation model, both local and long-range interactions take place simultaneously. Recent work has called into question the universality of the idea that each amino acid sequence gives rise to just one structure for certain proteins, even under normal cellular conditions. What are two classes of proteins that can give rise to multiple different structures? One, intrinsically unstructured proteins. 2, metamorphic proteins. When do intrinsically unstructured proteins assume a defined structure? Only upon interaction with other proteins. What does this molecular versatility mean? One protein can assume different structures and interact with different partners, yielding different biochemical functions. IUPs appear to be particularly important in what type of pathways? Signaling and regulatory pathways. Metamorphic proteins exist in an ensemble of structures of approximately equal energy that are in equilibrium. What happens when small molecules or other proteins bind to a particular member of the ensemble? Results in a certain biochemical function that differs from that of another complex formed by the same metamorphic protein bound to a different partner. What are cytokines? Signal molecules in the immune system that bind to receptor proteins on the surface of immune system cells. What do they instigate? An immune system response. We now know that in some cases, a gene can encode a single protein that has more than one structure and function. Craziness. List some diseases that are associated with improperly folded proteins. 1. Alzheimer's disease. 2. Parkinson's disease. 3. Huntington disease. What do all of these diseases result in the deposition of? Protein aggregates, called amyloid fibrous or plaques. To these diseases are consequently referred to as amyloidoses. What is a common feature of amyloidosis? Normally soluble proteins are converted into insoluble fibrous rich in beta sheets. What are porins composed of? Largely or completely of a cellular protein called PRP, which is normally present in the brain. The priors are aggregated forms of the PRP protein termed PRP-SC. What does the structure of the normal protein PRP contain? Extensive regions of alpha helix and relatively little beta strand structure. Within PRP-SC, evidence indicates that some parts of the protein that had been in an alpha helical or turn conformations may have been converted into beta strand conformations. What model for the transmission of amyloidosis has consequently emerged? Protein aggregates built of abnormal forms of PRP act as nuclei to which other PRP molecules attach.